Thanos, as you and I were talking about yesterday, other features of the SUCCESSOR-2 trial include a significant number of patients who are POM refractory, and that kind of sets it apart. So this is clearly a very high-risk population. You know, as principal investigator, Thanos, other thoughts to add to Katja's beautiful summary? I mean, there are so many things about this trial that set it apart from what we've done in the past. Any other points to add?
Yeah, I mean, again, I would like to highlight that we are dealing and we will be dealing in the future with an increasing number of patients who are both LEN resistant, exposed resistant, and anti-CD38 exposed resistant. So this is the target population of the SUCCESSOR-2 study. And not only we saw an important hazard ratio of 0.48, indicating that there is a 52% reduction of the probability of progression or death, with a median PFS of 18 months in the Mezi arm and 8 months in the KD arm. We saw interesting activity in patients with high-risk cytogenetics. We had significant activity with a hazard ratio of 0.3 in patients with extramedullary disease.
Yeah.
And also, it is important to highlight that the patient's characteristics were such that heavily pretreated patients were included. About 25% of the patients had received more than 4 lines of therapy, and only 15% had received 1 prior line of therapy. So maybe the characteristics of this study are a bit different from many other early myeloma salvage trials.
No, I think that's beautifully put. And as a co-principal investigator with you, Thanos, what really struck me about it in terms of as we designed it together was this necessity to build phase 1 of the study and then phase 2. And just to explain to our audience what that meant, we actually had to engage in a very careful dose optimization strategy pursuant of guidance from the FDA. And I do think it really paid off, didn't it? From the phase 1-2 experience, we had a clue that the milligram dosing was going to be appropriate with the 3 weeks on, 1 week off from the original combination with carfilzomib, which actually generated a response rate of over 80%. But I think the dose optimization was very wise because you nailed it, Thanos. The population was complicated, wasn't it? There was a lot of prior therapy, there was multiple prior settings, and so this dose optimization phase was critical. So just to share with the audience what this involved, it was a 1 milligram dose level, it was 0.8, 0.6, and 0.3, and the whole point was that we then dovetailed into the best dose level that was selected.
1 milligram.
Exactly, and then brought in that whole population. So just in terms of the size of the study, it was over 600 patients total, and the group assessed for efficacy was close to 480. So this was, I think, a really powerful platform going forward in terms of assessing benefit. Katja, as you